Hello everyone, today we will discuss EREG, also known as prosecridin. This molecule is far more than just another growth factor ligand. It plays a crucial role in both cancer development and inflammatory processes. Today, we'll take you through its molecular mechanisms. We will be explored from 6 parts: abstract, expression and distribution, structure, signaling pathways and roles in cancer, mediates drug resistance and targeted therapies. So, what exactly is EREG? It's a ligand for 2 key receptors, EGFR, also known as ERB1 and ERB4. By binding to these receptors, EREG activates a cascade of signals that influence processes essential to health and disease, including inflammation, wound healing, angiogenesis, and cellular proliferation. EREG is primarily expressed in lymphoid tissues and bone marrow. It's found in monocytes, macrophages, dendritic cells, and Langerhans cells. Importantly, its presence isn't limited to healthy tissues. It's also actively involved in shaping the tumor microenvironment, where it can influence immune responses and tumor behavior. Let's look at its structure. EREG is a single-pass type I membrane protein, consisting of 169 amino acids. It features an epidermal growth factor-like domain, and undergoes glycosylation to form a mature transmembrane protein. Through cleavage by metalloproteinases, it releases a soluble peptide that is biologically active and capable of initiating signaling. EREG plays a dual role in both signaling activation and tumor microenvironment modulation. Upon binding to EGFR or ERB4, it triggers dimerization and activates key pathways, such as RAS-ERK, PI3K-OCT, and JAK-STAT. These signals are transmitted via autocrine, paracrine, or juxtacrine mechanisms, affecting not only tumor cells but also stromal components like fibroblasts and macrophages. In the tumor microenvironment, EREG promotes tumorigenesis, for example, driving intestinal epithelial proliferation in colitis-associated cancer. Through the ERK pathway, it also induces the transformation of normal fibroblasts into cancer-associated fibroblasts via JAK2, STAT3, and IL-6 signaling, which in turn facilitates epithelial mesenchymal transition and metastasis. Furthermore, EREG enhances cancer stem cell properties and is part of the lung metastasis signature in breast cancer, collaborating with MMPs and COX-2 to promote distant spread. Perhaps one of its most clinically relevant roles is in therapy resistance. In non-small cell lung cancer, EREG secreted by tumor-associated macrophages can induce resistance to EGFR inhibitors like erlotinib. In colorectal cancer, it mediates 5-FU resistance. And in breast cancer, it contributes to tamoxifen resistance by altering cellular metabolism through the Warburg effect. The good news is that EREG is now a target for drug development. Several promising agents are in clinical trials. Li3848575 is a monoclonal antibody against EREG, currently in phase 2 trials for chronic neuropathic pain. Another is fepixnibart, a bispecific antibody targeting both TGF-A and EREG, also in phase 2 for osteoarthritis pain. These represent new avenues for precision therapy. That's all for today. More on drug targets. See you next time.